Hi, my name is Cameron, and my thesis is about the effect of exosomes on cystic kidney cell proliferation. So first, just some background. The disease we're looking at in particular is PKD, which is autosomal dominant polycystic kidney disease. It is the most widespread inherited kidney disorder in the world. It's really interesting because it's caused by just a single mutation in one allele of either PKD1 or PKD2, one of those two genes. That mutation causes the formation and growth of renal cysts throughout one's lifetime and then results in eventual kidney failure in 50% of all cases. In particular, we're looking at the interaction between exosomes and PKD, or exosomes are extracellular vesicles. They're very important for cell signaling, for material transport between cells. And some suggest that also they have an effect on APKD progression, where cystic cells are kind of, exporting exosomes that are taken up by non-cystic cells, and that kind of influences their behavior to make them more cystic in turn. Research has shown that these exosomes from renal epithelial cells do actually have these two proteins that are mutated in PKD. And a paper by Ding et al. Showed that if you take exosomes from PKD1 mutant mouse renal cell cultures and incubate them with normal mouse renal collecting duct cells, you actually see decreased PKD1 and PKD2 expression in those cells and also increased cell proliferation, suggesting that there is kind of a cystic effect. They also show that exosomes taken from 80 PKD patient urine have a very similar effect on the mouse cells. There are still a couple questions that are unanswered though, which is first, do exosomes from 80 PKD patient urine also promote proliferation in human renal cells? Because all the past studies have been on mouse cells mostly. Also, what about renal cells that already have that PKD1 mutation? So cells that serve as kind of a model for 80 PKD patients themselves. That leads to the hypothesis of my thesis, which is that in normal and PKD1 mutant human renal epithelial cells, the urinary exosomes from 80 PKD patients will promote greater cell proliferation than the exosomes taken from normal individuals here. To test this, we have two phases for our experimental design. First, we need to isolate and characterize those urinary exosomes, and then we need to actually do the experiment and assess the effect of the exosome treatment on the cell proliferation. To start, we isolate the cells, and that's just sequential centrifugation. So we add some protease inhibitors to fresh urine samples to stop them from being degraded. We centrifuge them at 4,000 Gs to pellet out the debris. We supernatant, centrifuge that at 10,000 to pellet the large proteins we don't care about. And then we collect that supernatant and then ultracentrifuge that at 150,000 Gs. We pellet that will give us the actual exosome pellet, which we can collect those pellets, resuspend them in PBS, pass them through a filter to sterilize them. Once we have those isolation solutions, we can characterize them. First, we do that by quantification. When we're doing that is through nanoparticle tracking analysis, which is where we are passing these exosomes through these isolation solutions through a nanomicroscope, and it counts them, all the particles that it sees, and gives them kind of a size of the particles that it sees. And we're seeing the nice big peak right where we expect the size of exosomes to be that we're looking for. We also can do a BCA assay to look at the total protein mass in our isolation solutions. And we see that in these like 100 ml urine samples from 280 PKD patients and normal individuals, we're isolating a little bit more exosomes or a little bit more total protein from the 280 PKD patients than the normal patient. And that is kind of inconsistent with past research that shows that 80 PKD patients actually have more exosomes in their urine. Once we quantify them, we want to do a protein detection by Western blots to look at specific proteins. So we look at exosome markers, CD9 and CD81, just to make sure that we actually have exosomes in our isolation solutions, and we are seeing that in all of our samples. And then we want to look at the PKD1 and PKD2 proteins, which are supposed to be in these exosomes. And we have done a lot of different blots and a lot of different antibodies, and so far we've been unsuccessful in this. We're getting like either huge streaks that don't really correlate with the sizes or other problems. And so obviously that's a big problem that we're having that we need to work on. We're not getting successful blots for those proteins. Another problem that I didn't really mention before is that those total protein quantifications that we're getting are a lot higher in mass than we're expecting if based on like the particle numbers of exosomes that we're seeing. One potential explanation that we have for this is that we're failing to separate those exosomes from uromodulin. That's the most abundant protein in urine, and so that makes sense that it could be inflating our total protein value. It's also a very sticky protein, especially when it's polymer form, and so it could be trapping those exosomes and their contents and preventing them from being successfully analyzed. And so we're going to look at seeing if that's the case, and if it is the case, then we're going to work on a way to separate those and then get pure exosomes. Once we do that, we can move on to our second phase, in which we're treating the cells with the exosomes and looking at the proliferation. We're measuring their proliferation by two different ways. First, an XTT analysis, and that we just take 296 well plates, one with PKD mutant cell cultures and one with wild type cell cultures, and where all of the wells have an equal number of cells seated in them at the very beginning. Then we treat them with exosomes, either no exosomes or exosomes from 80 PKD patient urine or exosomes from control urine. And we let them go for a while, and then we incubate them with XCT reagent, which is going to only incorporate into live cells. And then this so that we can measure the absorbance and see, okay, how many cells do we have in each of these wells? And if we do multiple different time points, then we're hoping to get kind of a graph like this, where it shows we have like kind of growth curves that differ between our different groups. And the way we can do that is flow cytometry. It's a very similar process. We have a plate of seeded with PKD1 mutant cell cultures and a plate seeded with wild type cell cultures. We treat them with having no exosomes, 80 PKD exosomes or control exosomes. We'll also have a third plate for like staining controls that don't have any exosomes in them. And then we're going to incubate them with EDU, which is when we're incubating it, it's only going to incorporate itself into DNA that is being actively synthesized. And so if we stain the EDU, it's going to give us those cells that are actively proliferating. And then if we stain all DNA, we're going to get a sense of all of the cells that are, that have DNA in them. And so hopefully after that, we can get, information like this, where we're going to have particle area and width, and we can get the, we can gate for this specific section, which is single cells that aren't clumped up or anything. And then we're going to be able to see what proportion of them are actively proliferating from this EDU fluorescence. And then also we can get kind of a graph to see what proportion of cells are in each phase of the cell cycle to kind of look at how these exosomes treatments are affecting the cell cycles of the different cell cultures. So yeah, those next steps, obviously we need to improve our exosome separate isolation. If it turns out that we are having uromodulin contamination, then we're going to try adding a sucrose gradient centrifugation step. Which basically pellets out all of the uromodulin while giving us a nice band, suspended band of the exosomes that we're looking at. And then maybe we might also need to adjust our Western blot protocols because there's some research that suggests that PKD1 and PKD2 are very delicate, like they're very big proteins, they form complexes, they aggregate sometimes, and so maybe our Western blot protocols might be a little bit too harsh for them as of right now. And then once we have those and we have fully isolated exosomes, we're going to perform those exosome treatments on the appropriate cell cultures. We've done some preliminary analysis, but it has been mostly on immortalized cell cultures. And the problem with immortalization a lot of times is that it's not very good for assessing actual changes in proliferation because the immortalization process makes them proliferate so differently already. And so we're hoping to look at iPSC cells that we're about to start differentiating to look at a more appropriate model for assessing proliferation. This is my acknowledgements. I'd like to thank my lab, Dr. Chapman and Dr. Chen, and also Dr. Wesley for a lot of advice along the way, and the Jackson Food Foundation for funding my research.
We have time for questions.
Yeah.
How would you make the Western like less harsh?
Looking at just like exposing them to lower temperatures when we're about to like pass them through the Western blot, when we're incubating them with like the reducing agent, maybe like lowering it a bit because we're seeing that maybe they might be degrading at the temperatures that are usually used for Western blots. Things like that, maybe using a less harsh reducing agent, etc.
Is there any variability in your samples?
We have a lot more samples than just these two. We haven't seen that much intervariability, especially with the exosomes that we're isolating. We haven't done like a full protein analysis on all of them. We're trying to make sure that all of the patients that we're collecting urine samples from have the same, like a truncating mutation in PKD1 especially, so that they're in the way that affects PKD, because there's some difference in like which gene's mutated and how it's mutated, that at least they're consistent in that.
I think this is part of what your illustration is, but what does how do people think PKD1 mutations affect like the contents of exosomes?
The idea is that PKD1 and PKD2, if either one of them are mutated, they affect just like a lot of different aspects of cell metabolism, especially, and also like adjacency of cells, their recognition, because PKD1 and PKD2 form a complex on the membrane of a cell. And so the idea is kind of both these two proteins are being packaged and exported to other cells, and also just other materials that have been kind of impacted by these changes in the cells are being brought into cystic cell or non-cystic cells that don't have these problems, and this is kind of changing the behavior of the cells by introducing these other proteins that have problems.
So like, what are the expected like even try to change when you're treating yourself?
We are expecting to see, and the main thing that we're looking at, like we're also looking at like changes in cell cycle, et cetera, but the main thing that we're looking at is just proliferation. That is the biggest hallmark of like these cystic cells, is that they're proliferating faster. And so that is what we're kind of looking at, is that if we treat cells with exosomes with these mutations, then they're going to start proliferating more.
Just more out of general curiosity, if you're posted on there, is there any reason why it's choose about eating using instead of like amorphous like cases?
No, not really. I think it's just what we have in our lab at the moment, and I haven't seen much evidence that it would be, bad for this kind of experiment. But yeah, we're definitely keeping our minds open as we're, about to go into this process of like what we're actually going to be using.
Did you say that the mouse, maybe you said this, the mouse exosomes actually have an impact on the human cells?
No, other way around. That human exosomes from human urine are having an impact on mouse cell cultures.
So the human exosome protocol has been worked out so people have
Yeah.
successfully isolated instead of just getting it going in early.
Yeah. There's a lot of exosome isolation from urine is just, it's not very standardized as of right now because it's such a booming thing that there just hasn't been a lot of like really standardization of the best way to do this.
And I was just wondering if it was somehow easier to do it in mouse than a human, or if it's just getting the protocol
Not
going
as
in early.
not as much research takes it from urinary exosomes. A lot of it is like growing cell cultures and then taking exosomes from that.
I see.
And that's has a lot less contaminants, but it's also a lot less accessible and so much slower. Yeah.
Thank you.